Functions of vertebrate circulatory system. Circulatory system is not simply a transporting system alone. It has several other functions also. Some of its functions are the following: transport of nutrients from the sites of their absorption to the sites of their utilization for oxidation, assimilation, and storage. Transport of O2 and CO2. O2 is transported from respiratory surfaces or organs to tissues, and CO2 from tissues to respiratory organs. Then transport of metabolic wastes from tissues to excretory organs. Transport of informational molecules like antibodies and hormones from their sources, production centers to target sites, action sites. Transport of metabolites from one tissue to another for further metabolism. For example, transport of glucose from intestine to liver for glycogen synthesis. Transport of lactic acid from skeletal muscles to liver for oxidation. Transport of toxins from tissues to kidneys or other excretory organs for filtration and elimination through urine. Supply of necessary substances to secreting glands. Then defense against infections and toxins by releasing antibodies and also by the phagocytic activity of some leukocytes. Then regulation of body temperature by virtue of the high specific heat, high latent heat of vaporization, and high thermal conductivity of blood, and also by its high powers for heat distribution through kick flow. Then maintenance of a proper and constant body pH, acid-base balance, maintenance of water balance, and the homogeneity of tissues by transporting excess water to excreting glands. Then regulation of osmotic potential and electrolyte balance by transporting surplus salts to excretory organs. Then storage of water and some electrolytes such as glucose, NAK, etc., which are constantly needed for tissues. Then kinds of circulatory system. There are three major kinds of circulatory systems among animals. They are blood vascular system, lymphatic system, and water circulatory system. Blood is the transporting medium in the first one. Lymph is the transporting fluid in the second one, and water is the transporting fluid in the water or a watery fluid is the transporting medium in the third one. Blood vascular system is found in annelid arthropods, mollusks, and chordates. Lymphatic system is found in vertebrates, and water circulatory system is found in sponges, coelenterate, helminths, and echinoderms. In some organisms, pseudocoelomic fluid is the transporting medium. Annelida, blood contained mostly in closed blood vessels, closed beries. Arthropoda, blood or hemolymph contained mostly in hemocoel, sinus, and lacunae. Mollusks other than cephalopods, same. Then cephalopod, mollusks, and primitive chordates, blood contained mostly in closed blood vessels. And vertebrates, blood and lymph contained in closed vessels, closed circulatory system. Water circulatory system, porifera, sponges, water in the canal system, coelenterata, water in the gastrovascular canals, platyhelminthes, watery fluid in the branching alimentary canal, and nematodes, pseudocoelomic fluid in the pseudocoel. Nemathelminthes. Echinodermata, water in the water vascular system. Blood vascular system is of two kinds, namely open blood vascular system and closed blood vascular system. So open system. This is the blood vascular system in which blood flows mostly through sinuses and lacunae. Sinuses are open channels with a membranous lining, whereas lacunae are open spaces without a membranous lining. Sinuses and lacunae merge together and form a large blood-filled space called a hemocoel. The blood that fills the hemocoel is called a hemolymph. Open or lacunar type of vascular system is found in arthropods and mollusks, with the exception of cephalopod mollusks. In open circulatory system, RBCs are absent, so oxygen-carrying respiratory pigment remains dissolved in blood plasma. This is the condition in the closed circulatory system of non-chordates also. In open circulatory system, blood flows with low pressure and low velocity. Pressure very seldom. So it takes a long time to complete a circuit in the body. Some terrestrial snails and some bivalve mollusks are exceptions in having high blood pressure. This high blood pressure results from the contraction of heart in snails, but from muscular contraction in bivalves. The high blood pressure and open circulatory system is significant in that it helps the animal to maintain posture. Since the blood flows most of the time through sinuses and lacunae, most internal organs are directly bathed by blood. Consequently, the exchange of nutrients, especially gases, wastes, etc., occur directly between tissues and blood. In open system, the flow of blood is not properly regulated anywhere, so it is relatively less efficient than the closed system. Then closed system, this is the circulatory system in which blood always flows through closed blood vessels. It is found in some annelids, earthworms, cephalopod mollusks, sepia, oligoctopus, and chordates. In the closed blood vascular system of non-chordates, RBCs are absent and hemoglobin is in the blood plasma. But in the closed system of chordates, RBCs are present and hemoglobin is seen in them. In closed system, the flow of blood is regulated at various points, so the system is more efficient than the open system. In the closed system of vertebrates, blood flows from arterial to venous circulation through narrow capillaries. This capillary network allows fine control on the distribution of blood and also causes the blood to flow under high arterial pressure. This high pressure is provided by the pumping action of the heart. The capillary wall is extremely thin and highly permeable. This permeability and the high arterial pressure allow high rates of exchange of materials between blood and tissues and enable the filtration of fluid from blood to intercellular spaces. This fluid is returned to venous circulation via the lymphatic system, which is a low pressure tissue drainage system. Thus, the closed blood vascular system for vertebrates is characterized by extensive capillary network, high arterial pressure, and the associated lymphatic system for recycling the fluid that leaves out to the tissue spaces. Need to learn a comparison or differences between open and closed systems. Open system, blood flows mostly through sinuses and lacunae. RBCs are absent. Oxygen carrying pigment is dissolved in blood plasma. Large hemocoel present. Blood flows with low pressure and low velocity. Blood takes a long time to complete a circuit. Internal organs are directly bathed by blood. Exchange of materials occur directly between blood and tissues. Supply of materials to tissues and removal of waste from tissues are very slow. The flow of blood is not regulated anywhere, so the system is relatively less efficient. In closed system, blood flows mostly through closed blood vessels. RBCs are present. With the exception of non-chordates, oxygen-carrying pigment is seen in RBCs. Hemocoel absent. Blood flows with high pressure and high velocity. Blood takes only a short time to complete a circuit. Internal organs are not bathed by blood. Then exchange of materials between blood and tissues occur through capillary walls. Supply of materials with elimination of wastes are very quick. The flow of blood is regulated and hence the system is more efficient. In closed system, we learned all these important points. Then significance of closed blood vascular system. Closed blood vascular system brings about a quick supply and rapid removal of substances. This is because blood flows under high pressure and high velocity and completes its circuit within a much shorter time. In closed system, the flow of blood is precisely regulated as per the needs of tissues. Next is about division of closed circulatory system. The circulation of blood through closed blood vessels was first discovered and described by William Harvey in 1578 to 1657. The most sophisticated type of closed circulatory system is found in vertebrates. It has two divisions, namely cardiovascular system and lymphatic system. The former is composed of blood vessels and heart, and the latter consists of lymph vessels and lymph heart. Blood and lymph are the circulatory body fluids which transport and distribute materials within the body. Blood contains carrier molecules such as hemoglobin for transporting large amount of O2, CO2, etc. Next, vascular fluids. Among animals, 6 types of vascular fluids can be recognized, namely pseudocoelomic fluid, coelomic fluid, hemolymph, blood, tissue fluid, and lymph. Pseudocoelomic fluid serves as a vascular fluid in pseudocoelomates, nematodes, endoprocs, rotifers, etc. And coelomic fluid is the vascular fluid of eucoelomate invertebrates, annelids, and echinoderms. This bring about the distribution of nutrients to different parts and the removal of nitrogenous wastes from different parts. They contain floating and wandering cells known respectively as pseudocoelomocytes and organic and inorganic substances but no respiratory pigment. Hemolymph is the fluid found in the hemocoel of some invertebrates such as arthropods and mollusks. It is regarded as equivalent to the blood and lymph of higher forms. Hemolymph contains proteins and wandering cells called hemocytes. Hemolymph has respiratory, circulatory, and excretory roles. It distributes O2 and nutrients to all parts and removes CO2 and nitrogenous wastes from different parts. Tissue fluid, also known as the intercellular fluid or interstitial fluid, is the clear intercellular fluid and with the low protein content, some salts, nutritive materials, and wastes. In invertebrates which lack circulatory system, it contains primitive and largely phagocytic cells. Also, it surrounds the body cell and forms minute lakes. Invertebrates, tissue fluid is a part of the blood and it is leaked out from capillaries to intercellular spaces under high hydrostatic pressure. It is gradually returned to the circulatory system by a system of channels called lymphatic system or lymphatics. Lymph is the part of the tissue fluid that has entered into a system of branching tubes called lymph vessels or lymphatics. Terminally, lymphatics drain the lymph to veins. Proper lymphatics is absent in invertebrates, cyclostomes, and cartilaginous fishes. In some bony fishes and amphibians, tissue fluid and lymph proper are sometimes in direct continuity. Next is about composition of human blood. Blood is the brief transporting medium in the human body. In a healthy male, there will be 5 to 6 liters of blood, and in a healthy female 4 to 5 liters. Blood constitutes nearly 8% of total body weight. Human blood is a thick red colored and slightly alkaline pH 7.3 to 7.45. Viscous fluid. It consists of blood plasma and blood capillaries. Plasma is a fluid part. Corpuscles are the blood cells loosely suspended in the plasma. Plasma forms about 55% and corpuscles 45% by volume. Plasma weighs as much as 5% of the total body weight. The instrument used for the measurement of blood plasma and blood corpuscles is called a hematocrit. Its measurements are called hematocrit readings. Blood plasma is a slightly alkaline yellow fluid. About 90 to 92% of it is water. The rest consists of organic and inorganic compounds in solution and suspension. Plasma water has three major functions: transports substances in a dissolved or suspended condition, regulates body temperature, regulates blood pressure and blood volume. Organic constituents of blood plasma. Blood plasma. Blood plasma is a slightly alkaline yellow fluid. About 90 to 92% of it is water. The rest consists of organic and inorganic compounds in solution and suspension. Plasma water has three major functions: transport substances in dissolved or suspended condition, regulates body temperature, regulation of blood pressure and blood volume, and organic constituents of blood plasma. The organic compounds of blood plasma include proteins, non-protein compounds, secretion products, and wastes. The major plasma proteins include albumin, globulin, fibrinogen, thrombogen, that means prothrombin, and heparin. Albumin and globulin in general help to retain water in blood plasma. This in turn maintains the normal viscosity, pressure, and volume of blood. They also transport substances such as thyroxine, Fe++, etc. Then albumins in detail. Albumins are water-soluble proteins. Albumins are water-soluble proteins which readily coagulate on heating. They form the major fraction of plasma proteins by virtue of the smaller size and large abundance of their molecules. They contribute nearly 80% of total osmotic potential of blood plasma. Albumins regulate blood volume, exert considerable osmotic pressure to maintain water balance between tissues and blood, and play an active role in the transport of lipids, hormones, bilirubin, naphthoquinones, sulfonamides, barbiturates, etc. Liver is the major center of albumin synthesis in the human body. Insufficient albumin synthesis causes a rare condition called analbuminemia, characterized by low serum albumin and low blood pressure. It has no serious clinical effects. Globulins. Globulins are coagulable proteins, that means they coagulate. They are of three major kinds, namely alpha, beta, and gamma globulins. Alpha globulins bind and transport thyroxine, bilirubin, lipids, steroids, and mucopolysaccharides. Beta globulin can bind cholesterol, lipids, metals like iron and copper, and the vitamins A, D, and K. The metal binding globulins are generally called transferins. An example is the copper binding globulin ceruloplasmin. The rate of synthesis of this protein is very low in Wilson's disease, like hepatolenticular degeneration, which is characterized by excessive deposition of copper in liver and brain and neurological manifestations similar to those of Parkinsonism. Gamma globulins are also called immunoglobulins. They serve as antibodies and play a vital role in immune mechanism. They are mainly concerned with binding antigens and histamines. So, a gamma globulinemia is a condition in which gamma globulins are absent in blood plasma. It results in the lack of immunity against bacterial infection. Then fibrinogen and thrombogen. Fibrinogen and thrombogen or prothrombin are the plasma proteins involved in blood clotting. During blood clotting, fibrinogen is covered to fibrin by the enzyme thrombin. So, fibrin undergoes polymerization and forms the clot, which helps the enzyme fibrin stabilizing factor. The blood plasma from which fibrinogen is removed for blood clotting is called the blood serum. Next, heparin. Heparin is an anticoagulant, anti-clotting factor that prevents thrombosis, blood clotting inside blood vessels. Non-protein organic constituents, secretions and wastes. Non-protein organic compounds include glucose, amino acids, fatty acids, glycerol, cholesterol, vitamins, etc. Secretion products include hormones, enzymes, antibodies. Waste products include urea, uric acid, ammonia, creatinine, xanthine, hypoxanthine, etc. Then inorganic constituents of blood plasma. The inorganic constituents or the electrolytes of blood plasma include mineral ions such as the chlorides, iodides, carbonates, bicarbonates, phosphates, and sulfates of sodium, potassium, calcium, magnesium, iron, copper, etc. They are essential for the maintenance of normal acid-base balance, pH level, ionic balance, osmotic potential, and the physiological balance between blood and tissues. They are also involved in the clotting of blood, contraction of muscles, A2+, transmission of nerve impulses, and strengthening of bones. Next is about functions of blood plasma, transport of substances that get dissolved or suspended in blood plasma water or get complexed with plasma proteins. Then maintenance of the normal volume, pressure and viscosity of blood by retaining water in blood. Maintenance of the normal volume, pressure and viscosity of blood by retaining water in blood. Maintenance of normal blood pH by neutralizing strong acids and bases. Regulation of body temperature through uniform distribution of heat all over the body. And conduction of heat to skin for dissipation. So blood corpuscles are generally called the formed elements of blood. Mammalian blood corpuscles are of three main kinds, namely erythrocytes, leukocytes, and platelets. Of these, erythrocytes are most numerous blood cells and constitute more than 98% of total blood cells. Non-mammalian corpuscles include erythrocytes, leukocytes, and thrombocytes or spindle cells. Thrombocytes differ from mammalian platelets in that they are nucleated. Platelets are non-nucleated. Mature corpuscles have no powers for division and multiplication. The total number of blood corpuscles per 1 milliliter of or 1 cubic mm of blood is called total blood cell count. The total number of blood corpuscles per 1 milliliter is called total blood cell count. Blood cell count is taken by the instrument hemocytometer. Blood cells are relatively short-lived. To make up their large-scale destruction, they are formed in large numbers almost instantaneously. Formation of blood corpuscles is called hematopoiesis or hemopoiesis. Formation of erythrocytes is called erythropoiesis. That of leukocytes is called leukopoiesis, and that of platelets is called thrombopoiesis. Then in mammalian embryo and fetus, they are formed mostly in yolk sac, liver, spleen, and red marrow. But after the fetal life, they are formed only in red marrow. So, schematic representation of the composition of human blood plasma 55%, water 90 to 92%, organic constituents 80 to 9%. Organic constituents in the sense proteins, albumins, globulins, fibrinogen, thrombogen, heparin, about 7%. Then non-proteins, glucose, amino acids, fatty acids, glycerol, cholesterol, vitamins, etc. Then secretions, hormones, antibodies, enzyme, bile salts, bile acids, wastes, urea, uric acid, ammonia, and creatinine, etc. Then inorganic constituents in the sense 0.8%, chlorides, iodides, bicarbonates, carbonates, phosphates, and sulfates of sodium, calcium, potassium, magnesium, iron, copper, etc. Then dissolved gases, oxygen, carbon dioxide, and nitrogen. Then blood corpuscles, 45% formed elements are also there. Erythrocytes, 4.5 to 6 million per cubic millimeter of blood. Leukocytes, 5,000 to 9,000 per cubic mm of blood. Granulocytes, basophils 0.5 to 1%, eosinophils 2 to 4%, neutrophils 60 to 70%, agranulocytes, lymphocytes or monocytes. Then platelets, 2 to 4 lakhs per cubic mm of blood. Next, in detail, we are going to learn about erythrocytes. Erythrocytes or red corpuscles are the small and the most numerous blood cells. The average total count of human blood RBC is 4.5 to 6 million per cubic mm of blood. However, it may go higher in the evening, after meals, during muscular exercise at high altitudes, high environmental temperatures, high barometric pressures, and so on. Also, it is high in infants during the first few days of life. Usually, it is higher in males than in females. The pathological condition in which RBC count goes abnormally high, 10 to 15 million per cubic mm of blood, is called polycythemia. The opposite condition in which RBC count of the hemoglobin content of the blood falls abnormally low is called anemia. Mature mammalian erythrocytes are non-nucleated circular disc-like biconcave bodies without endoplasmic reticulum, mitochondria, Golgi bodies, and centrioles. However, non-mammalian erythrocytes are nucleated cells. Young mammalian RBCs are nucleated, but as they mature, they lose their nucleus and membrane-bound cell organelles. A mature erythrocyte consists of a spongy network known as stroma. It is enveloped by a limiting plasma membrane. Stroma is formed of interlacing lipoprotein molecules. Its lipid constituents mainly include lecithin and cephalin. Stroma contain hemoglobin and carbonic anhydrase and many other enzymes. About 280 million molecules of hemoglobin in a mature red blood cell. Drainage of hemoglobin from RBC is known as hemolysis. Hemoglobin is a red-colored iron-containing respiratory pigment involved in the transport of O2 and CO2. Carbonic anhydrase is involved in the transport of CO2. Biochemically, erythrocytes are formed of water, hemoglobin enzymes, organic constituents, and inorganic constituents. Nearly 100 enzymes are believed to be present in RBCs. Carbonic anhydrase, dehydrogenase, catalase, alkaline phosphatase, etc. are the major groups of enzymes. Organic constituents include proteins, lipids, urea, amino acids, creatinine, phosphoglycerol, and lecithin, cephalin, cholesterol, and cholesterol esters are the major lipid constituents. So, potassium phosphate is the major inorganic constituent, NaCl is the major inorganic constituents of blood plasma. The average lifespan of erythrocyte is 120 days. It is estimated that nearly 3 million RBCs die every second. To compensate this heavy loss, an equal or still greater number is produced in the bone marrow. Normally, the rates of production and destruction of RBCs are mutually balanced. The rate of production of RBC is determined by the oxygen level in the blood. If the oxygen level is low, bone marrow will produce more RBCs than those are destroyed in liver and spleen. It is an adaptation of mammals at higher altitudes. If the oxygen level is high, bone marrow will produce lesser number of RBCs. The production of RBC is controlled by a renal hormone called erythropoietin. Worn out corpuscles undergo breakdown in liver and spleen. During this, the globin protein part of this hemoglobin gets degraded into amino acids. The porphyrin molecules of the heme portion gets degraded into the bile pigment biliverdin, which eventually of the heme portion transforms to the yellow pigment bilirubin. Biliverdin and bilirubin serve as bile pigments. The iron atom of the heme part is stored in the liver in the form of an iron-containing protein called ferritin. It would be used again for the synthesis of hemoglobin or cytochrome. Next, functions of erythrocytes. Transport of respiratory gases O2 and CO2, maintenance of normal blood pH and ionic equilibrium, formation of bile pigments by the degradation of hemoglobin. And leukocytes. Leukocytes or white corpuscles are the largest and the least numerous blood cells. Their average total count is 5,000 to 8,000 or more per cubic millimeter of blood. The average lifespan of leukocyte is 4 to 12 days. Leukocytes are colorless and nucleated. Remember, they are nucleated. Amoeboid cells. They are mainly involved in the defense against infections. Some of them ingest and digest foreign matters and invading microorganisms. They are known as phagocytes. Some others produce antibodies to develop immunity against infections. So red blood cells, erythrocytes, surface view platelets, white blood cells, or leukocytes. Nucleus present, agranulocytes and granulocytes. Lobed nucleus is there in case of neutrophils, eosinophil, basionophil, and monocyte. Headset shaped nucleus is seen in eosinophil. There are two groups of leukocytes, namely granulocytes, granular leukocyte, and agranulocytes. Agranular leukocyte. Granulocytes are formed in myeloid tissue in red marrow, and agranulocytes in lymphoid and myeloid tissue. Granulocytes have granular cytoplasm and lobular nucleus, but agranulocytes have non-granular and homogeneous cytoplasm. There are three kinds of granulocytes, namely basophil, eosinophil, and neutrophils or polymorphs. Agranulocytes are of two kinds, namely monocytes and lymphocytes. Neutrophils and monocytes usually serve as phagocytes. There are two kinds of phagocytes, namely microphages and macrophages. Microphages are small neutrophils, whereas macrophages are large monocytes. These monocytes, which can migrate to infected tissues, are called wandering macrophages. They clean up cellular debris after an infection. Some animals in the infected and inflamed tissue attract phagocytes to the tissues. This is called chemotaxis. These chemicals include toxins and degradation of products of damaged tissues. Most leukocytes have the ability to crawl through the minute intercellular spaces in the capillary wall and also through connective and epithelial tissue. This is called diapedesis. Sometimes the total count of leukocytes fall abnormally low. This condition is called leukopenia. Similarly, it may go abnormally high sometimes, especially in malignancies, inflammations, and acute infections. This is called leukocytosis. In some rare instances, some parent cells of leukocytes become cancerous and they undergo uncontrolled multiplication. This leads to an abnormal overproduction of one or more kinds of leukocytes. Most of the resulting cells are immature and abnormal. This very serious condition is known as leukemia and or blood cancer. Then basophils. Basophils are granular leukocytes whose cytoplasmic granules stain deeply with basic dyes such as methylene blue. They comprise 0.5 to 1% of total leukocyte count. They move about in an amoeboid fashion and ingest bacteria. Often basophils leave the capillaries, enter tissues and release heparin, histamine and serotonin. They are also believed to be involved in allergic reactions. The abnormal high count of basophils is called basophilia and their abnormally low count is called basopenia. Then eosinophils. Eosinophils are the granular leukocyte whose cytoplasmic granules are large and they stain brightly or brilliantly with acid dyes such as eosin. They comprise 1.5 to 3% of the total number of leukocytes. Eosinophils are believed to compact allergens. They leave the capillaries, enter tissue fluid, and produce antihistamines which destroy antigen-antibody complexes. In parasitic infections, skin diseases, and some allergic states such as asthma and hay fever, their number goes much higher. This condition is called eosinophilia. Abnormal fall in the number of eosinophils is called eosopenia. It is common under conditions of stress and strain of the body. Eosinophils are involved in resisting viral diseases, helminth colonization, post-pubertal mammary gland development, then allograft rejection, neoplasia, etc. The number of eosinophils is believed to be regulated by a hormone in the adrenal cortex. Then neutrophils. Neutrophils are granular leukocytes, so cytoplasmic granules are very fine and they do not stain or stain only faintly with acidic or basic dyes. Their nucleus is polymorphic and many lobed and therefore they are also called polymorphonuclear leukocytes or polymorphs. They are able to squeeze out into the intercellular spaces through the pore spaces of the capillary walls. This is called diapedesis. Earlier we mentioned about this. Then from the intercellular spaces, they move to infected areas. Neutrophils exhibit amoeboid movement and are highly phagocytic. They can engulf and digest foreign particles such as bacteria using the enzymes from their granules.